Hello, everyone. Thank you for joining us today. My name is Allison Herrera, and I am the Content Specialist here at Alcami, and I welcome you to today's webinar, Alcami Introduces Rapid Sterility. Today's session will be recorded and made available to you in the coming days. If anyone has questions, though, along the way, feel free to type them into the Q&A section of your toolbar, and we'll be sure to answer those questions at the end of the presentation. At this time, I'm going to turn the floor over to my colleague, Senior Scientist, Stacey Ramsey, to begin today's webinar.
Thank you, Allison, and good morning, everyone. First, I want to thank all of you for joining me today. I hope the information we're providing will be insightful in some way for all of you. Today, I want to share with you a new offering coming from the Microbiology Laboratory at Alcami's Tech Center in Wilmington, North Carolina. This webinar represents the launch of a new technology for our organization, specifically an alternate test methodology to the Compendia Sterility test called the Milliflex Rapid Sterility test. First, we'll be introducing you to the technology platform I just mentioned, the Milliflex Rapid Sterility test. Next, we'll provide a detailed description of the technology and the methodology, and I'll explain how upgrading to a rapid sterility test provides benefits to the industry and to you, our clients. We'll provide a side-by-side comparison of the Milliflex Rapid Sterility test to the Compendia Sterility test as it's described in USP Chapter 71 and the Harmonized Compendia. And then finally, we'll wrap up by telling you what you should expect from Alcami should you choose to contract us to perform the rapid sterility test for your drug products. So what is rapid sterility? Rapid sterility is an alternate microbiological test method to the USP 71 test that allows for shorter incubation periods and faster results to clients. Rapid sterility has been around in the industry for quite some time. There are many technological innovations for rapid sterility. These, just to name a couple, are flow cytometry, ACP bioluminescence, laser-induced fluorescence, and there's probably many more that we've not mentioned here. Alcami has selected the Milliflex Rapid Sterility system, which is based on ATP bioluminescence. What can rapid sterility do for you? Rapid sterility can release your drug product to the market faster at QC release or even during stability. We can reduce your inventory holding time, and we can provide a robust computer-generated sterility result. This is based on software that is 21 CFR Part 11 compliant. And what's great about this system is it eliminates the subjectivity and individual analyst observation that's inherent to the USP 71 test as it's described. So just a brief description of the rapid sterility. Sample processing takes place in a hard barrier sterility test isolator. So the initial parts of it are not unlike a USP sterility test. Alcami's sterility program has a rigorous cleaning and monitoring program for the sterility test environment. So we're not treating our rapid system any differently than the Compendia test in that we are maintaining the strictest aseptic environment for our testing. Alcami does trend our EM data for our sterility test suite, and rapid sterility meets all the same QC requirements as the referee method. The rapid test incubates not less than 5 days, which allows us to offer you, our clients, a shorter turnaround time. And also, we will provide a more sophisticated computer-generated result in lieu of an analyst observation. The reports look something like this. What's great about these reports are that we have a very good depiction there of the sample. In this particular example, we have microorganisms on the sample. This was done as part of our comparability protocol, which included inoculation with microorganisms. If you can see there, the software also offers a colony count. So we don't have to visually depict those colonies and interpret them. The software does that for us. To describe this a little bit more, as I mentioned, the initial process is similar to the USP 71 test. Samples, materials, and the interior isolator chamber are subject to VHP decontamination prior to the test, just like a USP test. Milliflex uses membrane filtration via specialized vacuum pumps. If we have any microbiologists out there, you might all be familiar with the Milliflex system. This is not unlike the Milliflex system that is currently used in the industry for bioburden testing, for water testing, and that kind of thing. Millipore has specialized pumps that are VHP compatible that we have in our isolator chamber that we use for the sterility test. So it's slightly modified from the original Milliflex system. Samples are filtered through the filter funnels, followed by rinses as determined by a bacteriostasis fungistasis test, which is the method suitability for sterility. Filter funnels are removed, and funnels are applied to media cassettes, just like the original Milliflex system. Media cassettes are incubated at 3 conditions. We incubate a plate at 30-35, one both in aerobic conditions and anaerobic conditions. This mimics the FTM media that is listed in USP 71. We also incubate one of the cassettes at 20-25 C in aerobic conditions to mimic the incubation requirements of TSB media. One thing that we don't describe in this slide is we are using a rapid sterility test media. It is a blood agar media. It's the same media used at all conditions. Each condition is treated as a separate media as it relates to USP 71 tables 2 and 3. If you're familiar with these tables, they tell you how much sample you're required to process per media type. Because the original USP method has 2 medias, we have 3, so that would have to be taken into consideration when you consider how many samples you have to test. Media cassettes incubate for not less than 5 24-hour periods. At the conclusion of the incubation period, the analyst relocates the media cassettes to a sanitized biological safety cabinet for sample processing. And here's where things get a little different than the original Milliflex system. Membranes are separated from the media cassettes using a specialized tool. So we discard the agar media and we have left behind the membrane filters. Membranes are treated with 2 reagents for lysing cells and treating with a bioluminescent tag. Bioluminescent tag binds with ATP. Bioluminescent tag illuminates under the specialized camera. ATP is a proven indicator of cell viability. ATP is produced by all living cells in order to make energy, so we know that if ATP is present in the sample, we're looking at microbes. Milliflex is therefore a growth-based method that requires an initial incubation period, but it detects microorganisms before they can be observed with the naked eye. So here are a few examples. These were taken during our comparability protocol. This was the protocol that we used to compare the Milliflex rapid system with the USP test. This is incubation of Pseudomonas aeruginosa, which is one of the organisms listed in USP 71. And you can see how the colonies light up using the ATP bioluminescent tag. At the conclusion of the ATP bioluminescence procedure, we took that membrane and put it on a fresh agar media cassette and placed that back in the incubator. And this is what we got. So as you can see, for our side-by-side comparison, the colonies are in almost the exact same locations as what we saw during the ATP bioluminescence procedure. And in that way, we can directly correlate the growth to ATP bioluminescence. We also did this for a number of other microorganisms. We found that the anaerobic incubation condition works very well, as you can see for Clostridium sporogenes. We also studied the sterility focus microorganism, Propionibacterium acnes. If you're not familiar, this is considered one of the slowest growers in a sterility test and is one of the indicators for why we have a 14-day sterility test. Using the rapid system, the Propionibacterium acnes grows in about 4 days. So that's why we set our incubation period at not less than 5 days to allow for an extra day of buffer to make sure that we get everything. And then, of course, we also looked at environmental isolates. Environmental isolates are very important during comparability studies for a rapid system because the FDA wants to see that real-life microorganisms that could be present are detectable using the alternate system. The USP sterility test in our shop has about a 4 to 5 week turnaround time. So this includes all of the sample receipt, the login, the vetting of all of the materials as they come in, the setting up of the isolators, the 14-day incubation, and of course the QC release. The incubation period is probably our biggest rate limiter. The test itself incubates for not less than 14 days. It does require us to prepare an isolator for testing. An analyst observation is used to generate the results. So many of you are familiar with the canister test system. At the end of the test period, the analyst will pick up the canister, swirl it, and look for some cloudiness. In comparison, our Milliflex rapid sterility test, we're offering a 3-week standard turnaround to clients. A big part of that is the decreased incubation period. This also requires isolator use. Here at Tech Center, we've added a second sterility isolator to our laboratory, so we are also helping to turn turnaround time a little quicker there. We are also providing a more sophisticated computer-generated result. This is just a picture from the software. We get a 3D image and a 2D image, so we can look at colonies should we see them. Again, software removes subjectivity from the test when we're using the rapid sterility test. Software provides additional analyst controls to the test. So, for example, once I add a sample and I analyze the sample, there's no way to delete the data once it's created. There's no way to change the data. There's no way to change the label on the data. Software provides an audit trail so that we can see everything that's happening during the analysis of the sample. This is just kind of a quick anecdotal thing that's kind of neat. The rapid test does not use a peristaltic pump, so it's kind of better for shear sensitive products. This uses a vacuum filtration system, so we're not shoving your product through a peristaltic pump. During our comparison studies, we looked at the compendial monographs for statistically comparing our rapid sterility system to the referee's sterility method. We have 3 main articles here. USP 1223 is probably the most valuable during our comparability studies. Technical report number 33 was also very helpful. It provided a lot of additional information, but it's also very similar to USP 1223. Farm Euro does have a chapter for alternative methods. It doesn't have a lot of information in it, but we know that it's there, so we know the Farm Euro will be accepting alternate methods. They just don't give us a lot of direction for how to study the new methods. So here at Alcami, we performed a comprehensive comparability protocol demonstrating that the Milliflex rapid sterility system is equivalent to the referee sterility method. Alcami performed a most probable number study. So a lot of people who work in parenterals aren't entirely familiar with most probable number. The MPN method is a way to take a qualitative test. We test at multiple dilutions, and we assign a quantitative value to the number of colonies present in the original sample. So per USP 1223, we prepared cell culture suspensions to a known concentration. This is about 10 to 100 CFU, which is similar to what you would make for a bacteriostasis fungistasis test. Then we diluted out in 10-fold dilutions. We tested these dilutions in not less than 5 replicates, side by side, using both the USP sterility and rapid sterility methods. This is, for each microorganism, about an entire day of work. So it's a lot of work because we want to prepare one culture that's used to test side by side both methods to make them as comparable a study as we possibly can. We determined the most probable number from 3 dilutions in series that provided positive and negative growth, or in the case of the MilFlex system, signal. We studied 13 microorganisms. USP 1223 requires us to study the 6 compendial microorganisms that are listed for our chapter, in this case, USP 71. We also studied 6 environmental isolates. 3 of these were from a manufacturing environment, and 3 of these were from the laboratory environment. And of course, we studied P. acnes because we know that the FDA likes to focus on this one as a slow grower. We generated MPN and 95% confidence intervals for each one. During our study, we found that 12 of 13 microorganisms showed equivalent recovery for the 2 methods. 1 microorganism, which was an environmental isolate, showed better recovery using the MilFlex rapid system. So for the most part, what we see is equivalent recovery. In one case, we saw a little better recovery. We also performed a statistical non-inferiority test. This test is described in USP 1223. That test demonstrated that the MilFlex rapid system is equivalent to the compendium method as a whole. So if you were to come to Alcami and ask us to perform a rapid sterility test for your product, we want you to know what we would provide for you. Alcami will perform method suitability for your drug product. That's the bacteriostasis fungistasis, to establish an appropriate method for your specific product. We have a number of documents that we would provide for you so that you can have these for your application filing. The vendor, Millipore, has a validation report that they used to qualify the system as a whole prior to selling it for use. We also have our comparability report, which is required for alternate methods. Then we would also issue to you your method suitability report. So you would have all the documents that you need for your filing. Also, Alcami has subject matter experts in MilFlex rapid technology who will support regulatory questions as they arise. Alcami also has established a strong working relationship with Millipore when additional support is needed. So what we'd ask of you, our clients, is if you contract us for this test and you have a filing, we would definitely like to know all about the feedback we're getting from the FDA. This is a new thing for the industry for the last several years, and people are still kind of learning, and we'd like to know, too, what kind of feedback everybody is providing. So with all of that, the brief description, that's kind of all we have for you today. I do want you all to know that even though we didn't go into the in-depth scientific discussion of comparability with the compendia method, we will be offering that in the future. We will also be present at the PDA Micro Conference in Bethesda in October, where we will be presenting on the science of the comparability protocol. We'll have a booth there, and if you're there, we'd love to meet you and talk. We will also be at PharmSci in San Antonio in November.
Thank you, Stacy. Also joining us today for our Q&A is our Director of Microbiology, Angela Harrison. And it looks like we've had a few questions come through during the presentation, but feel free to continue to type in your questions as we go along. Okay, so for our first question we got, why did Alcami decide on the ATP bioluminescence platform versus the flow cytometry or laser-induced fluorescence as our rapid sterility platform?
That's a good question. So the reason that we opted for ATP bioluminescence is that it works well for regrowth. So in your sterility test, you want to have a path forward should you need to perform an investigation for a sterility failure. If we do have microorganism recovered on the samples, as I showed you, we can regrow those and then perform a genetic ID of anything recovered so that we can aid in the sterility investigation.
Thank you, Stacy. Should we expect more investigations or organisms with the new computerized data evaluations versus a subjective methodology?
So during our comparability study, we performed statistical equations that were meant to show how our new system compares to the USP compendia test. what we found during that comparison was that they were very comparable. Only in one case did we actually have better recovery. So we would not expect that we would see more contamination or more sterility positives with a rapid test versus the compendia test.
Thank you. Next question we have is, does rapid sterility require more samples as compared to the USP sterility test, as there are 3 growth media to evaluate?
Yes, in this case, for this method specifically, we would need 1.5 times the amount of sample as a compendia test because we have to incubate each condition using the table requirements in USP 71.
Okay, thank you. 1 second. Does rapid sterility by ATP bioluminescence also facilitate genetic identification?
Yes, and that was part of our comparability studies. There's also publications in some PDA literature that this has been studied as far as the regrowth procedure and how good it is at recapturing growth. So in our cases, we were able to get microorganisms to regrow so that we could then do a genetic ID test. That's what makes this probably the best rapid sterility system in my personal opinion.
Okay. Has the rapid sterility test been approved by the FDA for a USP 71 alternative?
So we would need for every drug product to have its own submission of the rapid sterility test. Now, having said that, this rapid sterility test has been accepted by the FDA in cases out in the field. We are launching this for the first time at Alcami, so we do not have any approvals here, but this system does have approvals out in the field.
Okay. Have any of your clients filed this rapid sterility for NDA or ANDA filing?
Not yet. So this webinar is the introduction of our use of Milliflex into the field, and we are not at that point yet where we have any approvals just yet.
Okay. Does Alcami also do the standard 14-day culture alongside the 5-day rapid sterility test?
So there would not necessarily be a need to do that. It can be done if that's requested. The USP does say that in the case of any kind of, if we're unsure of the result, the USP method is the referee method, and that result would stand. If that's what clients would like us to do, we can certainly do it, but having shown comparability, it wouldn't be something that would be required.
Okay. Is rapid sterility harmonized across EP and JP?
So, as I mentioned earlier, we have an EP, a Farm Bureau chapter, that broadly discusses alternate methodologies. Now, there is no specific methodology implied by the chapters in the compendia. Only the USP chapter tells us how to demonstrate comparability. So I guess to answer that question, there is no specified method. We make the method, or Millipore made the method, and we are contracted to do it, or we will be contracting it as well. But there's no, since it's not a specific specified method, it can't be a harmonized method. I'm not sure how to say that.
I mean, that's what the comparability protocol is showing for us, is that it is comparable to a harmonized method.
Yeah.
Okay. Has this method been submitted to CVM or CDR?
So I am not exactly sure what branches have accepted this method. I do know that the FDA in general is fairly familiar with the Milliflex rapid system, but I do not know what branches have approved the method for use.
The software calculates the microbiological levels. And if working in a sterile facility, this is an interesting technology. What is the typical precision?
So if I understand the question right, the millipore validation does say that the limit of detection is 1 CFU. So I don't know if that helps with understanding precision specifically. For qualitative methods, we are not required to analyze precision. This is required for quantitative methods, but the limit of detection is validated as 1 CFU.
Okay. Next question is, can we get a copy of these slides? I can go ahead and answer that. Yes, a copy of these slides will be made available to you in the next coming week. And are there any more questions? Feel free to type them in. We'll give you a couple minutes. Okay, thank you all for your great questions. If you think of any more questions later on, or if you'd like more information, or would like to meet up with Stacy at one of the upcoming events, you can email her at stacy.ramsey@alcamynow.com, which is also shown on your screen. Also, as I mentioned at the beginning and just recently in the last question, this webinar has been recorded, and you will receive a follow-up email in the next few days with a link to the recording as well as a copy of these slides. And with that, we'd like to say thank you for taking the time to join today's webinar, and have a nice rest of your day.